Next up is intracellular accumulation. So there's a bunch of different types of intracellular accumulations that we're going to go over. So where do these substances come from? Well, they can either be synthesized by the affected cell itself, or they can come from elsewhere. Where do these accumulate? It kind of depends. So they can accumulate in the cytoplasm, within the organelles, or inside the nucleus. It kind of just depends on the type and the process for that specific accumulation. So how does this happen? This varies, but there's 4 main pathways of abnormal intracellular accumulation. So we're going to go over each of those. So the first is if you have inadequate removal of a normal substance secondary to some sort of defect in the mechanisms of packaging and transport. So the example here is showing you this is your normal cell, and then if you have this abnormal accumulation of fat inside the liver. So this is a liver cell, and you just have a lot of fatty little blobs kind of accumulating inside the cytoplasm. So this is an example of fatty change, also called steatosis of the liver. The second pathway that this can happen, if you have an accumulation of an abnormal endogenous substance, so from created by you. So this would be a result of some sort of genetic issue or otherwise acquired defects in the folding, packaging, transport, or secretion. So there's a lot of different things, and we'll get into some of these when we get into the genetics chapter. But one example of this, you don't need to know this yet, but just to give you an example, is one type of a mutation of alpha-1 antitrypsin can cause different accumulation of abnormal proteins, which is that antitrypsin inside the actual cell itself. So that's one example, but again, you don't need to know that yet. We'll get to that when we get to that. The third way that you can have something accumulate in the cell is if you fail to degrade some sort of metabolite due to an inherited enzyme deficiency. So these are classically referred to as storage diseases. There's lysosomal storage diseases and other types of storage diseases that, again, we'll get to later on. This is just to give you an idea of how these things can happen. And then the last way that this can happen is if you have some sort of exogenous substance, so not made by that cell, and it's coming into the cell. And so if the cell doesn't have the enzymes needed to degrade it, or if it doesn't have the ability to transport it elsewhere, then it can accumulate within the cell itself. So an example of this would be an accumulation of carbon, and we'll see that in a little bit. So these accumulations, can this be fixed? So if these accumulate inside the cell, can you get rid of them? It kind of just depends. Sometimes this is reversible, and sometimes it does lead to cell death. So it depends on the type and the severity of the accumulation. So the types of accumulations, we just went through the 4 ways that this can happen. Now we're going to go over the 4 basic types of these accumulations. So the 4 main types, you have lipids, proteins, glycogen, and pigments. So we're going to start with lipids. Now, lipids, you can have 3 different types of lipid accumulation. So you can have pretty much all the major classes of lipids. So you have triglycerides, you have cholesterol and cholesterol esters, and then you have phospholipids. So you can also get, there's those lysosomal storage diseases that I just mentioned. Then you can have weird abnormal lipids accumulating. So there are exceptions to this, but the general rule is that these 3 types of lipids can accumulate within the cell. So we'll go through examples of each of these. So the first example, if you have triglycerides, so that was the first one I mentioned. So triglycerides, if those are accumulating, that is what we call fatty change or steatosis. And this is classically going to be found in the liver because that's the major organ that is involved with fat metabolism. But it can be caused by toxins, protein malnutrition, diabetes, obesity, and other types of causes. So we're going to go through a couple different examples, and we'll start with the liver because, like I said, that's the major organ involved in fat metabolism. So if you have a healthy liver and you have some sort of abnormal fat metabolism or some sort of other problem going on, you can get accumulation of those triglycerides within the liver tissue itself. And so that will cause fatty liver or steatosis of the liver. Then over time, that can progress to liver fibrosis. Why is this not working? There we go. That can progress to liver fibrosis, which can then lead to cirrhosis. So for now, we don't need to know these things. We're just trying to understand the fatty liver. But that's why fatty liver is bad. And we'll talk about that later on when we get to the liver chapter. But basically, your healthy liver has some sort of problem that can cause fat to accumulate within the hepatocytes themselves. So here's an example of that. So here's a normal liver, and then there's the two most common causes of fatty change in the liver, or fatty liver disease, are alcohol abuse and non-alcoholic fatty liver disease, also called NASH, N-A-S-H. And NASH is typically associated with diabetes and obesity. So that's why in developed nations, these are the two most common types or causes of fatty change in the liver. So oftentimes you'll see basically these big blobs, and as we learned in histology, these will show up as clear on normal H&E slides. So here, all of these are just little accumulations of fat, and same thing over here, just like a bunch of fat. And see how it's kind of like intermixed with the hepatocytes? So that is showing you fatty liver disease. Now, these are fairly normal hepatocytes. You don't see a lot of fatty cells or fatty accumulations within the cells. So this, like I said, is classic for the liver, but it can also happen in other organs such as the heart. So here you can see there's a lot of fat on this heart. Now this is just fat on the outside of the heart, but I just wanted to give you a gross visual. So histologically, what is going to happen is these are your cardiac myocytes, and I know it's hard to see because you have all these pigments here. So just note these pigments for right now. We're going to come back to this later. Just note that this is lipofusion inside these, and we'll get to that when we get to the pigments. But you can also see, like within these branching cardiac myocytes, you can see some accumulation of fat as well. And so you can get some triglyceride accumulation within the actual heart tissue itself. Another organ that can get this is kidney. So this you should recognize as kidney tubules, but these do not look like normal kidney tubules, right? So you can see that all of these epithelial cells in the kidney tubules, you can see tons of fat accumulation within those. And here's another example. So this would be more zoomed out. You can see all of these accumulations of fat, and you know this is kidney because you can see those glomeruli, right? And you can see some normal looking tubules, like that looks like a normal-ish distal convoluted tubule, but all of these have fatty change. Same thing over here. This is just a zoomed in view of this area so you can appreciate it a little bit better. So you can see that these are accumulating within the different cells. Ooh, that's a really good one right there. They're accumulating within these epithelial cells inside the kidney. So that is the triglycerides. Now moving on to the cholesterols and the cholesterol esters. So the classic example of this is atherosclerosis. So in atherosclerotic plaques, you have smooth muscle cells and macrophages inside that intimal layer of the vessel. And typically this is going to be the large and the medium-sized arteries, right? So in that layer, that's going to be filled with lipid vacuoles, which are made up of this cholesterol or the cholesterol esters. So basically all of this is going to aggregate inside the intima, and that's what produces that yellow appearance grossly of these plaques. So this is just a cartoon here, but you can see it grossly too. So here's kind of how athero happens. So you have this LDLs coming in. It's going to go inside that intima, and then once it's in here, the macrophages are going to eat it up, right? Because the macrophages don't want that fat accumulating inside that intima. So you'll activate the monocytes. The monocytes are going to leave that vessel lumen, come through the epithelium to then enter the intima, and then they're going to become a macrophage, right? And then the macrophage is going to eat up those LDLs, and then over time they're going to look like this little foam cell, but over time they can actually rupture, and then the rest of that macrophage will undergo apoptosis, and all of those lipid little fragments are going to accumulate in the intima. And this appearance here, that's when you get the plaque, which is going to look yellow on gross examination. So take a note of this little foam cell here, because this is what you're going to see histologically as these intracellular accumulations. So once we get into the vessel chapter in systemic pathology, we'll actually talk about, the formation of the plaque and plaque rupture and all of that. But for now, this is just to demonstrate an example of intracellular accumulation. So note the foam cell. Now histologically, this is what you're going to see. So all of these, this is why we call them foam cells, because on normal H&E, they look foamy. So all of these are foam cells, which are just those macrophages filled with that fat. And something else you might see histologically on a plaque is this. So this big kind of almost like cleft here, this is a cholesterol ester. So this can crystallize in the shape of kind of like a needle, and it forms this cleft here. So that's what this is. So all that is a cholesterol ester, and then all of this inside these macrophages, inside these foam cells, that's all fat accumulations within the cells. So that's an example of that. Another example is xanthomas. So cholesterol can actually accumulate inside the macrophages like we just saw, but if you have some sort of acquired or hereditary hyperlipidemic state or disease, then this isn't going to just happen in plaques. This can happen kind of everywhere. So it can happen, typically it'll be found, you'll have these like foam cells in the subepithelial connective tissue of the skin and also in tendons, and it will kind of create these little like tumorous masses. So here on this hand and the eyelid over here, these yellow masses, those are xanthomas. And so if you took a section of that, you would just see a bunch of foam cells, so those macrophages filled with fat underneath the skin. And then here, this is it right here. This is an example of a xanthoma kind of within the Achilles tendon. So that's what that is right there. And you don't see that like bright yellow appearance because it is deeper. It's inside the tendon versus over here, this is just deep to the skin. So a xanthoma is another way that these cholesterols can accumulate inside cells. And what you're going to see histologically, this is one from the skin. So this is your normal keratinized stratified squamous epithelium of the skin. But if you look down in the dermis, you can see, over here's good ones. These are basically those foam cells again, right? And if we zoom in on that over here, you can see that all of these, see those little foam cells? Those are all great examples of intracellular accumulation of those lipids, of those cholesterol fragments. And yes, those are still lipids. Another example is cholesterolosis. So cholesterolosis is an accumulation of these cholesterol-laden macrophages in the lamina propria of the gallbladder. So it's kind of unknown how this happens, but typically it's going to have something to do with a high cholesterol diet. And what happens is histologically all of these foam cells accumulate, but grossly what you see is this. You'll see this diffuse yellow stippling is how I would describe this gallbladder mucosa. And so if you say that in a gross description, typically that's telling the pathologist, I see cholesterolosis, you should see foam cells histologically. So then once they read your gross and they pull up their slide, this is typically what they're going to see. So you can see the gallbladder mucosa here, and then deep to that, or not the mucosa, this would be the epithelium, and then deep to that in the lamina propria, which is still part of that mucosa, right? You'll see these lipid-laden macrophages, aka this is cholesterolosis. So cholesterolosis, you'll see the kind of foam cells, and then grossly, you'll see the diffuse yellow stippling. And then lastly, those phospholipids. So we did the triglycerides, that was the fatty change or the steatosis. We just did the cholesterol and the cholesterol esters. And remember that third type of lipid was your phospholipids. So phospholipids, there's not much I really want you to know about these other than the fact that they can create these myelin figures in an injured cell. So whether it's reversible, like in this one, or irreversible, if you see those myelin figures, they're actually made up of those phospholipids. So in summary, for the lipids, we did the triglycerides and we talked about steatosis and fatty change, and our examples for that were liver, heart, and kidney. We talked about cholesterol and cholesterol esters, and so our examples for that were atherosclerosis, plaques, xanthomas, and cholesterolosis of the gallbladder. And then lastly, those phospholipids, we said that those can form those myelin figures. So that's it for the lipids. Now moving on to the proteins. So what proteins can accumulate within cells? So histologically, basically, this is just going to show up as these little round eosinophilic droplets, or they can sometimes show up as vacuoles, or sometimes it'll just be like little aggregates of stuff in the cytoplasm. So there's not one very obvious appearance, and there's also not one obvious cause. So there's a lot of different causes of this that we will discuss later throughout the course, but I'll give you just one example for now. So the example for now is the kidney. So in physiology, you learned about the kidney and what each part of the nephron does, right? Well, in the proximal renal tubules, these are going to reabsorb some protein, right? And so if you have some sort of renal disease where you have protein loss in the urine, aka proteinuria, what can happen is that all of this protein, there's tons of protein coming through, and so your kidney tubules essentially like can't keep up. And so normally you have small amounts of protein going through the glomerulus that's reabsorbed in that proximal tubule. But if you have tons of protein leaking across that glomerular filter, aka proteinuria, basically you have an increased reabsorption of this protein into the vesicles, and that's going to show up in the proximal tubule. So this is what that looks like. So here, this would be, the PCT, the proximal convoluted tubule, and you can see that this does not look normal. I know it's kind of fuzzy, but if we zoomed in, you would see little pink eosinophilic droplets kind of within that cell cytoplasm, and that is accumulation of protein because you have this proteinuria. Now, this is reversible to a certain extent, so if the proteinuria is fixed or decreases or whatever, that protein inside those proximal convoluted tubules can be metabolized and disappear. So it is technically reversible at this stage. Now moving on to glycogen. So where does glycogen accumulate inside the cell? So normally you have glycogen in the cells, right? So think back to like biochem, and what do we use glycogen for? So that would be for anaerobic respiration, right? And so normally you have glycogen stored in the cytoplasm of healthy cells. But if the patient has some sort of glucose or glycogen metabolism problem, you can get excessive glycogen accumulating inside the cell. So what does this look like? It kind of looks like the fatty change, honestly. It's a little bit hard to tell the difference, but we'll learn that as we go through the course. So this is an example of a kidney, and you know that because you can see the glomerulus, right? So kind of ignore this part for now. We're not getting into the details of this, but this is an example from a patient with diabetes. And so if this patient has glucose problems, which you do when you have diabetes, basically you can get the accumulation of this glycogen within the renal tubular epithelial cells. It can also accumulate in parts of the pancreas, part of the heart, and also liver cells. But classically in the kidney, you'll see these kidney tubules kind of filled with glycogen. All of these are filled with glycogen here. So basically it's like this vacuolization in the tubular cells. It's not like the very obvious kind of blobs that we saw before when you had the lipids, the cholesterol's accumulating, or sorry, not the cholesterol, the triglycerides accumulating inside the tubules. So you can kind of tell the difference already. Another way that this can happen besides diabetes is if you have a storage disorder. So within the storage disorders, which we'll talk about when we get into genetics, there's some that are called glycogen storage diseases, and you can actually accumulate glycogen within various types of cells. And then lastly, the pigments. So there are two main types of pigments, and pigments are just colored substances, right? So the two main types are exogenous and endogenous. So we're going to talk about the exogenous first. Now, I got two examples of this for you. So the first one is carbon. So carbon is just a normal pollutant in any urban area, and when you inhale this carbon, the macrophages inside your lungs are going to pick it up, and then it's going to bring it to the lymphatic channels, which then go to the lymph nodes surrounding the lung and the airway. So it's grossly going to appear as this. So this is just the outside of a lung, and you can see all of this black pigment this is anthracosis. I would write that down. So if you are inhaling this carbon, it will appear as black discoloration, and it is called anthracosis. Now, if you cut into the lung and it's pretty severe, you can see like big areas of black discoloration or anthracosis. So here's another example. This is like a perfect lung. It's a beautiful example of normal anatomy. And even this one, even though I'd say this is a fairly healthy lung, you can still see some areas of anthracosis. And that's because this isn't just going to be in smokers. This is, like I said, just a normal pollutant in any urban area. So typically, pretty much everybody is going to have some level of anthracosis. And actually, I found this study, which I think is really interesting. It's in the bottom right if you want to look it up. But this is comparing smokers versus non-smoker lungs. And I mean, you can still see that the non-smoker has some anthracosis, but I thought their findings were really interesting. So some of the highlights were that you can use this pleural anthracosis as a potential index of lifetime exposure to air pollution, which I thought was interesting. But they were talking about that if somebody commutes to work and sits in traffic, their carbon exposure is going to increase. And so even in non-smokers, if it's somebody who commutes or sits in traffic a lot, they'll have an increased amount of that anthracosis. It's also associated with increasing age, because the longer you live, the more, pollutants you're exposed to, and also smoking load, obviously. But the really cool part about this study that I found was one of their findings was that 1 hour of exposure to air pollution is equivalent to smoking 5 cigarettes a day. And I thought that was crazy high, which is really, really bad. Maybe we should not pollute the air so much. But yeah, interesting study if you want to look it up. I mean, I'm not going to ask you that on the test or anything, but I just thought it was interesting. So that's what it looks like grossly. But what does anthracosis look like histologically? For once, it actually looks fairly similar, which is nice. So histologically, it's also just going to appear as this black discoloration. So basically, you just have accumulations of this black pigment in the lung and the surrounding lymph nodes. So this is lung. It doesn't look like normal lung at all because it's very fibrosed and there's some inflammation and you have the anthracosis. But it is a section of lung. Now, this is a section of a lymph node. So over here, these are anthracotic lymph nodes. So you'll see black discoloration of the lymph nodes. And so if you ever get a lung specimen, like a lobectomy or even a total pneumonectomy, they'll typically send some lymph nodes with it as well. And typically they'll appear, maybe not this obviously black, but they will almost always have some sort of black discoloration. And even in the anatomy lab, when we pull out some of those lymph nodes, typically a lot of them are black because, like we just saw, even people who don't smoke are constantly exposed to that carbon. So if you take a section of a lymph node that's anthracotic, it's basically just going to look very similar to that lung in that you have all of this black discoloration. So you can see it just deep to the capsule, you can see it in the cortex and part of the paracortex, and you can see it throughout the medulla of that lymph node. So carbon was the first example of an exogenous pigment. The second example of an exogenous pigment is tattooing. So when you get a tattoo, they're basically putting this pigment inside your dermis, so deep to the epidermis, and the macrophages inside that dermis are going to come and eat up that pigment. And then the macrophages stay there for the rest of your life, which is why tattoos are permanent, right? I mean, there are some ways to get rid of them now, but for the most part, they're permanent. So here's an example of a tattoo, and then over here, this would be, your normal keratinized stratified squamous epithelium. Now we're in the dermis down here, and you can see all of this black pigment. Now moving on to the endogenous. So one is lipofusion. So lipofusion, remember earlier on that heart slide, I said to take a note of that because we'd come back to it. Well, we're coming back to it. So this is just this insoluble pigment. It's also called the wear and tear pigment. It's pretty much just a normal sign of aging. It doesn't cause any cell injury. It doesn't impact the cell's function. So typically you don't really consider it being important, but because it's there, it's telling you that person did have some sort of free radical injury. And that's because typically the lipofusion is going to form through lipid peroxidation of these lipids from the subcellular membranes. So they're basically made up of lipid polymers and phospholipids kind of admixed with protein, and that's why it's insoluble. But the term lipo is referring to fat, right? And then fuscus is referring to brown. So this literally just means like brown lipid. And so the reason they call it that is because histologically it's going to appear as this yellow to brown, finely granular cytoplasmic, but sometimes perinuclear pigment. So where are you going to see this? This is going to be, we called it the wear and tear pigment. So this is going to be in cells that are undergoing slow regressive changes. So this is going to be particularly prominent in the heart and also the liver of older patients or younger patients that have some sort of problem, so like cancer or malnutrition or something like that. But typically it's just in older patients, and it's just showing you like, oh, this person is older. So in this picture you can see that it's in the cytoplasm, it's kind of around the nucleus. All of this is just lipofusion. So here's some other examples of it. So these two on the left are heart, and remember we saw that on that previous heart too, and it was way more diffuse on that other one. But these are just your cardiac myocytes, right? And then in here you can see inside these cells, you can see that this lipofusion has accumulated within the cells themselves. Same thing here. So you can see that the lipofusion is inside the cell. Now this on the right, this is an example of liver. So here you can see that there's a lot of lipofusion kind of diffuse throughout all of these different hepatocytes. Another type of endogenous pigment is melanin. So we talked about melanin in histology. Remember, that's made by the melanocytes, which are located in that basal layer of the epidermis. So here, this is a really good example of the color of melanin, and notice that it is in that basal layer. So all of this is melanin pigment. And you should remember, hopefully, from histology how this forms. So remember, tyrosinase is going to catalyze the oxidation of tyrosine, and that's going to create melanin, and then the melanin can kind of go up through the melanosomes into the upper layers, which is why you're seeing some of it up here. Another example of an endogenous pigment is hemosiderin. This is going to be important when we get into systemic pathology. Hemosiderin you're going to learn about a couple times, but for now you just need to know generally what it is. So it's derived from hemoglobin, hence the name hemosiderin, and typically it's going to show up as this like golden yellow to sometimes brownish granular pigment, and it is the major storage form of iron, and that's why it has to do with hemosiderin, right? So if there's a local or a systemic excess of iron, ferritin is going to form these hemosiderin granules. So an example of a local excess is shown in this picture. So these are from the lung, and this is showing pulmonary congestion, and so these capillaries are dilated, which is why this area is so pink. But because these capillaries are dilated, that's bringing in a lot of blood into these lungs, and so that can cause, that's why we're calling it congestion, right? So the blood is kind of just sitting in these lungs. So if the blood is just kind of sitting there and the lung is congested, that can cause leakage of the blood into the alveolar space. So if that happens, the macrophages are going to come in and they're going to try to eat up all of that blood, and that will result in these hemosiderin-laden macrophages. So in the lung, these are sometimes called heart failure cells, because if you have heart failure, we'll get into all the different types of heart failure, but for now, just understand that if you have heart failure, one of the possible effects is that blood can kind of sit in the lungs. So if the left side of the heart isn't pumping blood out as well as it should, it has nowhere to go except backwards, right? So it will retrograde flow back into the lungs. And so if it's kind of sitting there in the lungs, you can get these heart failure cells, which are these hemosiderin-laden macrophages. So both of these are lung. This is a zoomed out view of the lung, and you can see all of these are different macrophages, and then all of the pigment inside those macrophages, that's all hemosiderin. And this is just a zoomed in view of the same thing. So all of this like golden yellow to brown kind of granular accumulation inside those cells, that is the hemosiderin. So that was one example of a local excess of hemorrhage or blood. Now, another example of a local excess of blood or hemorrhage is going to be a bruise. A bruise is a really good example of how this actually forms. So when you have a bruise, basically what happens is you're going to have hemorrhage in an area, right? It's literally just a hematoma. So blood cells, red blood cells, should only be in blood vessels, right? But if you have hemorrhage in an area, those red blood cells have now leaked out into the surrounding tissue. And so they're not supposed to be there, right? So macrophages are going to come in and try to eat up those red blood cells. The macrophages are then going to break the hemoglobin down to recover the iron. So it'll break that hemoglobin down into heme and iron. So after removal of the iron, the heme is converted to biliverdin, which is the green bile, and then to bilirubin, which is the red bile. But as that's happening, the iron is released from the heme. And so ferritin, it's going to be incorporated into ferritin, and that will eventually be made into hemosiderin. And so you can see all of this yellow discoloration of a bruise as it's healing is literally showing you what the underlying mechanism is. So as the heme is incorporated into ferritin to form hemosiderin, you have this greenish yellow discoloration here. And then also, since the biliverdin is, green bile, then that's also playing some sort of role in this too. So bruise is a good example of where you would find hemosiderin. Now, you can also get systemic overload of iron. So the lung example and the bruise example were examples of locally increased iron, but if you get systemic overload of iron, you can get hemosiderin kind of all over the place in lots of different organs and lots of different tissues. And when that happens, that's called hemosiderosis. And so there's a couple different ways this can happen, which we're going to learn when we go through systemic pathology, but I'll give you kind of a preview now. So if you have an increased absorption of iron, dietary iron, what can happen is if you're eating this iron and you have some sort of error in metabolism called hemochromatosis, you're going to have increased absorption of that dietary iron. So then you can get a systemic overload of hemosiderin. Another way this can happen is various types of anemias. So there's a lot of different types of anemias. Some of them are hemolytic anemias, which means that the red blood cells are being broken down, and so you're increasing the amount of iron that's being released. And we just saw how that forms hemosiderin, right? And then you can also have, if you have a lot of blood transfusions, then those red blood cells that you're transfusing, those are going to have extra iron, and so you can get a systemic overload of iron then too. And so, this can happen in a lot of different tissues, but this example that I have on the screen is from the liver, and so you can see lots of hemosiderin inside these iron cells, or not these iron cells, these hepatocytes. Okay, so in summary for the pigments, we talked about exogenous pigments, so we talked about carbon, and we talked about tattooing, and then we went into the endogenous pigments, and so we talked about lipofusion, that's that wear and tear pigment, and then we talked about melanin, which we've already talked about in histology, and we talked about hemosiderin, which is iron, right? So definitely understand the various types of pigment and where you might see them and why. And then there's also something called Highland Change or Highland Change, and this isn't really a pigment, but we like to group it with the pigments because it's like a discoloration, I guess. But Highland Change it's this homogeneous kind of glassy pink appearance, and it's not a pigment. It's just a descriptive histologic term. So it's not really like an accumulation of pigment inside a cell or anything like that. It's more so just like a morphologic feature of various types of things. So it's not really specific to anything, but I wanted to include it because you'll probably hear that as we move on in general and systemic pathology. So here's some examples of it. On the left, we have liver, and so you can see that these cells are very pink and homogeneous looking, and so that would be Highland Change of the liver. And then here, this is a very zoomed in view of a glomerulus, and so all of this would be Highland Change as well. And so as we move through systemic pathology, you'll learn various times you might see this, but I wanted to include it. So it's not really a true accumulation of pigment, but it's more so just a descriptive term. So that's it. Let me know if you guys have any questions on that.